Thanks, Gloria, and thanks for introducing the session and the Research on Tap series. It's just great to see people back on campus right next to each other after 2 years. And thank you, everybody, for coming to this event. I have just a few things to say, and then we should get on with the event. As you will see from today's talks, there's a lot of energy and research on the topic of human brain with an eye towards understanding how the brain processes and stores information. Today's event nicely aligns, as Gloria said, with the launch of the new center, and the Center for Brain Recovery is focused on detecting and identifying neurological disease, understanding the mechanisms that sort of pervade the different neurological diseases, evaluate which treatments might work for different kinds of people, and really focus on the patient's success as they continue to live their lives. Now, what sets apart the Center for Brain Recovery, and I think you'll see that in the talks today, is the focus on this interdisciplinary work. It's bringing together scientists, clinicians, and engineers, all addressing the same problem, the same complex problem of why the brain goes wrong when it does. And another important thing that sets apart the center from other similar centers is the focus on the patient. And I think that's really important these days, as you see almost every other day in the news, where very famous people are getting diagnosed with neurological disorders we always knew about, but now there's a renewed attention towards these questions. And I think you'll see some of that come through today, but that's definitely a big focus of the center as well. So I hope you'll get to enjoy some of the work that really highlights and shows the alignment among folks across the 2 campuses at Boston University addressing these questions. So without further ado, let's get started. I think our first speaker is Rob. Welcome.
Thanks, Swathi. It's an honor to speak in this forum. So in my lab, we are interested in the brain mechanisms of human cognition, and we're trying to build novel neuroscience therapeutics to help people with brain disorders. We're also interested in age. That's the focus of this 5-minute talk. So it's an unfortunate fact of life that as we age, we become more forgetful. And sometimes that becomes devastating with debilitating disorders like Alzheimer's disease. We know a tremendous amount about the brain networks that support our memory capacities, and there's studies that suggest the rhythmic activity of the brain may be an important element in those networks for coordinating information processing in the brain. What we need now are novel tools to non-invasively and selectively manipulate those networks to potentially allow us to determine whether we can protect or even improve memory in older people. So we have a capacity limited working memory system that allows us to hold information in the here and now. We have a capacity unlimited system, long-term memory system that allows us to hold information over longer timescales. There's nearly a century of research demonstrating that the primacy and recency serial position curve effects index these different memory capacities. So if I read you a list of words and you recall as many back as you can, you tend to be quite well for the early words in the list. That's the primacy effect linked to long-term memory. You also happen to be, you're quite well at the latter words. That's the recency effect linked to working memory. In terms of the brain, a number of brain regions are involved. For example, the dorsolateral prefrontal cortex and the inferior parietal lobule are major players in contributing to primacy and recency, working memory and long-term memory. What we don't know is whether there are rhythmic mechanisms within these segregated cortical regions that we can identify. If we can identify these rhythmic mechanisms, we may be able to use tools like transcranial alternating current stimulation to boost memory in older people in a rapid and long-lasting manner. And that's what we set out to do. So we conducted a series of experiments, randomized, double-blind, sham-controlled experiments in 150 older adults, 65 to 88 years old. We developed these novel high-definition transcranial alternating current brain stimulation protocols guided by electric field modeling. Happy to talk about the methods afterwards. And the overall objective was to use low frequency stimulation in parietal cortex and boost people's recency or working memory, to use high frequency stimulation in prefrontal cortex and boost people's primacy or long-term memory. People got a dose of 20 minutes every day across 4 consecutive days, and we looked at 1 month follow-up as well. And indeed, we found that we can boost your working memory, your recency effect, by applying low frequency, but not high, to parietal cortex. And whereas we can boost your long-term memory or primacy effect with high frequency prefrontal stimulation. But that wasn't the case when we applied low frequency. So there's spatial and spectral specificity across these first 2 experiments. We reproduced the result in a 3rd experiment with an independent cohort of individuals. But another primary finding was up here, where the rate of benefits over this 4-day intervention predicted how big the memory improvements were at the 1 month time point, 1 month later. And then finally, we also found that these older individuals who come into the lab with poor cognitive functioning, they're the ones that are benefiting most. If you look at the rate of improvement over the intervention, as well as if you look at 1 month after. And so we're also now looking at whether these effects transfer to other experimental paradigms, how long they last, and if we can port this over to a proper clinical trial with people with Alzheimer's disease. Thanks.
Hi, everyone. I'm Matt Nelson. I'm a assistant professor. I'm starting a lab at the University of Alabama at Birmingham. I recently received a KO1 award from the NIDCD. And for that award, Swati Kiran kindly agreed to serve as a co-mentor. So that's why I'm here today. I'm visiting her lab to learn from her and to learn about aphasia and to learn about the lives of persons living with aphasia. I'm going to present to you today research that I conducted before starting my lab, and which is research that I'm following up on in my lab. My graduate research involved working with monkeys and performing neurophysiological recordings of monkeys. And it was starting my postdoc was when I wanted to get into the human intracranial field. And starting that field, deciding what I should study in the human intracranial field led me to pose myself the question, what is it that you can study in a human that you can't study in an animal? And that led me to language, which is one of the quintessential features of humanity that separates us from animals. And although all animals communicate, what's unique about human language is, among other things, it's a unique capacity to apply nested rules in a hierarchical fashion. And to describe what I mean by that, I'll ask you all to consider the sentence, "The car at the stoplight is blue." Now, any English speaker can hear that sentence and they know that what is blue is the car and not the stoplight. And we do that even though the phrase, "the stoplight is blue," is present in the sentence in that order. However, we don't interpret the sentence in that order. Rather, we correctly interpret the phrase "at the stoplight" as being on a different level of the hierarchy of the sentence. In this case, it's an adjective phrase modifying the noun car. So this shows that a linear representation of words is insufficient to describe human language, and rather hierarchy is needed. So to study that hierarchy, together with colleagues, I conducted the study here that I'm going to attempt to review for you. So this is using the human intracranial technique. These are recordings that are made in patients with epilepsy that volunteered for our experiments. These are patients that were undergoing invasive monitoring as part of their clinical treatment. So while they're undergoing this monitoring, they're waiting in a hospital bed for about a week with electrodes implanted, waiting for seizures to happen. So this is fertile time for a cognitive neuroscientist and systems neuroscientists like myself. So we put a laptop in front of them and they perform our task while we get this access to these unique recordings. So we're looking at using the hospital's equipment to record what the brain is doing while they're performing our task. So for this task, we use a rapid serial visual presentation task. The patients are reading sentences presented one word at a time. And then at the end of the sentence, they're presented with a brief probe sentence, and they have to answer a question about the probe sentence. The specific task and the probe sentence aren't important for the study. We use them just to keep the patient honest, and it allows us to say for sure that the patient was attending to the stimulus. But what we care about in our analyses is the neural activity during the sentence. So that's what we'll analyze. In particular, what's of particular importance is the neural activity at these merge operations, which occur at the end of phrases. So that's what's shown in the upper left pointing arrows there. So those are the key moments for our analyses. If you consider that first phrase, 10 sad students of Bill Gates, there are 6 words in that phrase. It's at the end of the phrase where there are merge operations that happen. So what was 6 separate items becomes one item, and that becomes the item, the node higher in the hierarchy of the sentence. That's the subject noun phrase of the sentence. So that's the key time points that we analyze, and we present the patient with different sentences of different structures. As for example, the sentence shown at the bottom there has a drastically different structure than the top sentence. And we analyze specifically the broadband high gamma power, which has been shown by previous work to be an excellent measure of the overall activation of a population of neurons around the electrode recording site. So I'll give you a brief view of the example activity from one example electrode, which is here in the superior temporal sulcus in the left hemisphere. So for sentences that have the first merge that start at the second word, they would start with something along the lines of 10 students. So you see here the neural activity rises and then peaks following the merge operation and drops to a lower level later on. We can compare that to sentences that have the first merge at the third word. Those sentences would start with something along the lines of 10 sad students. You see the neural activity follows the same profile, peaking at a higher overall level after the merge operation and dropping to a lower level later on. We can compare that to sentences that have the first merge at the fifth word, and they would start with something like 10 students of Bill Gates. You see the neural activity, it rises again, peaks at the time of the merge and drops later on. And then finally, we can compare that to sentences that have the first merge at the sixth word, and they start with something like 10 sad students of Bill Gates, with the activity peaking at the phrase boundary and then dropping to a lower level later on. And this is found, that was one example electrode. This is found across electrodes. I'd be happy to discuss more details afterwards. Thank you.
Hi, everyone. Mike Esterman. I am in the School of Medicine, Department of Psychiatry, and at VA Boston Healthcare System. So when we're listening to these talks, or we're driving our car, we're reading an article, our attention isn't constant, but rather it fluctuates between periods of relative focus and periods of relative inattention due to mind wandering, distraction, or fatigue. And these fluctuations or lapses in attention, they have real-world consequences for our educational attainment, for our employment, our relationships, and our safety. And they also underlie and modulate many other, quote, higher level cognitive functions, such as memory and working memory. And they represent some of the most common cognitive complaints across a range of neuropsychiatric populations, coming in the form of difficulty concentrating, distracting thoughts, intrusive or ruminations, and dysregulated arousal. And this ability to sustain attention, it changes dramatically across the lifespan. It improves rapidly in development, where it may also be sensitive to our interaction with technology. And it declines with age and may be some of the earliest or most sensitive markers of neurodegeneration. So in my laboratory, we've developed a tool called the Gradual Onset Continuous Performance Task, or the GRAD-CPT. It involves these scenes that gradually fade from one to the next, and participants are asked to press to frequent city scenes and then withhold to these rare mountain scenes. And so the GRAD-CPT has been run in over 30,000 individuals around the world, in multiple laboratories across diverse populations, and can be deployed in a number of ways, including on the web. So on the upper left here is an example of a study we did on the web, where we characterized how this ability to sustain attention changed across the lifespan. And what you can see is that there's this inverted U-shaped function where there's this rapid improvement in ability in adolescence, and there's also age-related decline. And if you look at the bottom plot, what you can see is that there's a consistent relationship between impairments in sustained attention and self-reported disability. But further, this relationship strengthens with age. And this is consistent with some work on the upper right, where we find in multiple samples that in older adults, better sustained attention is associated with preserved healthy gait and mobility. And so we also find that this ability to sustain attention relies on the coordination between two large-scale brain networks. One, the dorsal attention network, which is important for goal-directed attention and behavior. And on the other hand, the default mode network, which is important for internal mental processes. And what we find in numerous samples, an example on the top left, is that using fMRI, individuals who are better at sustaining their attention on the y-axis show this greater anti-correlation or antagonistic processing between these two networks. We've also replicated this finding using intracranial EEG on the right in awake neurosurgery patients who are performing the grad CPT. And finally, we have some new and ongoing work trying to understand ways we can enhance or remediate this ability to sustain attention. For example, we're developing transcranial magnetic stimulation approaches to targeting the dorsal attention network to reduce attention lapses. On the upper right, we're collaborating to do some work using mindfulness meditation to potentially stave off age-related decline in sustained attention. On the bottom left, we work with an attention or cognitive training procedure in a range of populations, including Parkinson's disease and veterans with PTSD and TBI. And some of the most recent work we've been doing in collaborations with many people is to potentially develop ways to restrict mobile device use or social media use in younger populations, not only to improve attention, but potentially improve mental health outcomes. And so the hope is that, coming back to the first slide, that if we can intervene and find ways to improve sustained attention, we can improve things like mental health outcomes, safety, and people's daily functioning. So thanks so much for your attention.
Hello, I'm David Boas from BME, and I want to tell you about neuroscience in the everyday world using functional near-infrared spectroscopy. Actually, as my former postdoctoral fellow Alexander von Lumen likes to call it, NeuroX in the everyday world, where X stands for science or neuroergonomics. And the idea basically is we want to take portable, wearable neuroimaging technology, so not fMRI, and we want to study brain function in complex and dynamic and multi-sensory real-world environments to better understand the brain in its natural environment, but also to really relate that understanding of the brain with human performance in real-world tasks. And we need to take this from studying multi-trial analyses, as is typically done in neuroimaging, to bring it to the single trial level as well. So there's a lot of technology development there. And so we have we were very fortunate 2 years ago to get a nice brain initiative, UO1, and is bringing together a large group of ours in BME with a number of other faculty and their teams across BU to develop the technology and apply it. So we are working on developing a wearable whole head FNIRS imaging system, measure hemodynamic responses to brain activity. We're combining that with existing wearable EEG systems to get the neural responses more directly, combining that with eye tracking and other biosensors, all in a wearable package. And then working with about 12 different faculty and their teams around campus to then apply that technology to a number of interesting applications. And so this is a very interdisciplinary challenge in that, we're developing inexpensive technology that can cover the whole head with sensors while maintaining high quality performance. We're doing this multimodality, combining FNIRS with EEG, so we can combine neural electrical signals with hemodynamic functional responses. We have to develop the signal processing tools that allow us to bring that all together, all of that multimodality sensors together, and do that in a real world environment, very challenging. And then of course, there's all of the making all of that available to as broad a community as possible. We need not just the technology, but the tools for analyzing the data, standards for helping to share the data, cloud platforms, and the neuroethics of how to share that data, and then databases for all of that. So there's many challenges that we're all addressing. We have a great large group of here at BU. I wish I had a photograph of everybody. Swati, that's something we have to work on, so we could show that off here. But there's many more problems than we can all presently address. So if any of you are interested in any aspects here, I look forward to talking to you after this session this afternoon. So thank you.
Hi, thanks for the opportunity to let me talk about our lab's work with connectome fingerprinting and our efforts to extend this technique to neurosurgical patients. Connectome fingerprinting combines neuroimaging and machine learning to make high precision, individualized predictions of human functional brain organization. Potential applications range from defining highly efficient functional localizers for cognitive neuroscience to non-invasively assaying a patient's functional brain organization for precision medicine. Connectome fingerprinting uses measurements of the brain's connectivity to make high-resolution predictions of brain function. Dick Passingham conjectured that each cortical brain area has a unique pattern of connectivity, and if we could establish these patterns of connectivity and measure the connectivity pattern of a particular brain region, we could predict the functional identity of that brain region. That is, that brain structure predicts brain function. Connectome fingerprinting builds on this idea by incorporating non-invasive neuroimaging and greatly expanding the spatial extent of the predictions. To build a connectome fingerprint model, we first define a cortical parcellation scheme and identify a search space, a part of the cortex that we want to map and predict in high resolution. Then we need to identify a particular task that we want to predict and collect two sets of training data. We need fMRI task activation in this particular task contrast, and we need some sort of connectivity measurement. We can use resting state fMRI to compute a subject's functional connectome by taking the Pearson correlation between the time courses of each vertex in the search space with each cortical parcel. We then combine the functional connectomes and the activation maps in our machine learning approach to construct our connectome fingerprint model for that particular task. With the model in hand, we can make predictions for novel subjects of their task activation if only we have their connectome fingerprint. That is, all we need is resting state data on the novel subject in order to predict their task activation. We can validate the model if we also have the actual task activation for the subjects. We can compare the patterns of those and validate the model in a set of test subjects. And then once the model is validated, extend that to new subjects. We've had success in mapping and predicting working memory and attention function in parietal and frontal lobes. These are highly individualized predictions. Almost always the best prediction comes from using the subject's own functional connectome compared to using other subjects' connectomes or group averages. So this offers some clinical, a real clinical opportunity. Doing functional localizers is great, but you can't always do that. And in a clinical setting, you may not have a dedicated fMRI team available to collect this data, or your patient may not be able to do the tasks inside the scanner, but they could do resting state inside the scanner. Your regular staff could collect that data, and you could apply connectome fingerprinting to make these predictions in clinical situations. In our ongoing work, and this features great work by Vaibhav Tripathi, a PhD student in my lab, and in collaboration with Dr. Alex Golby and her team at the Brigham, we've been looking at extending these connectome fingerprinting techniques to pre-surgical glioma patients to predict their motor and language function organization. Our long-term goal here is to develop a tool that could be deployed to multiple neurosurgical sites and to be integrated with the existing neuronavigation systems that are used for pre-surgical planning and in the operating room. And the first phase of this, we took data from the Human Connectome Project data set for the motor and language tasks to construct our connectome fingerprinting models. We applied that to make predictions on healthy control in glioma patients collected at the Brigham. And with as little as 4 minutes of resting state data, which is much less than we did in the other experiments, we're able to get suitably decent predictions that at least demonstrate the feasibility of this approach. There are a lot of challenges that remain with this. In the next phase, we want to collect more resting state data per subject to get more accurate functional connectome estimates for individual subjects. We need to harmonize the scanner parameters and also more directly probe neurovascular decoupling that occurs in the vicinity of the lesion site. Nevertheless, we see a market opportunity here. Most neuroimaging neurosurgical centers lack the dedicated fMRI team to do this sort of mapping. And this could really be a much more cost-effective way to do functional mapping in individual subjects. So I want to thank my students, my funding agencies, and thank you for your attention.
All right. Hi, my name is Rhoda O. I'm from the Med Campus, and I want to talk to you a little bit about our Precision Brain Health Initiative. I like to focus on the concept of precision brain health as opposed to precision medicine. But having said that, we do have a worldwide problem when we look at the issue of dementia, and it is that this problem is actually increasing more in low to middle income countries than they are even in high resource countries. But here's the bigger problem. This is where the science is happening in the field. And as you can see, it's not happening in most parts of the world. So, there we go. So this is where we really need science to happen, right? We talk a lot about inclusivity. We actually need to figure out how to make that happen. And so the question is, where do we start? So for me, I focus a lot of my work on using digital, and particularly the smartphone, because it's the most penetrating technology in the world. And every smartphone has multiple sensors. Those are data collection tools. And you have computers in every person's hand. So there's a lot of things that you can do with digital that allows you to do it inclusively. And so this is what we're trying to do. So first you have to start where everybody else starts. So this is how we traditionally think about collecting data in the field, which is bringing people in, doing lots of different kinds of tests, but they tend to be very high burden. They take a lot of time, they require somebody to come in, and so this is what's leading to our bias research. We're moving in now into technologies. Most of them are active technology, active engagement. They require you to do something. They do lower the burden somewhat, but not actually enough. And so what we really need to head to is passive engagement technology, one that collects data continuously on an ongoing basis while you're just going about your daily life. And that's really the bigger vision of where we need to head. So I'm just going to tell you some of the work we're doing here at BU. I work with Framingham Heart Study, and we've been using a digital pen actually for our cognitive assessment since 2011. We actually introduced the concept of looking at digital voice recordings since 2005. Speaking is a very cognitively complex task, and most people speak. So one of the things we're trying to do is selectively test in a way that's maximum inclusive. And then we have now more recently moved into porting all these different kinds of cognitive assessments onto that smartphone. And then in addition, at BU, at our Alzheimer's Disease Research Center, we're kind of taking the same kinds of applications that we're doing at the Framingham Heart Study. We're adding to that some additional assessments that can also be done digitally in order to, again, expand sort of the base of how we look at cognitive behaviors, which are very, very complex and shouldn't be thought of as only being tested through neuropsychological testing. There are many, many different ways in which you can capture cognitively related behaviors. And then more recently, we've started the Davos Alzheimer's Collaborative, which was launched through the World Economic Forum in January of 2021. So we're taking the same kinds of mobile technologies, again, that are maximally available when you think about internationally and in low to middle income countries. We're taking all the same types of tests that we've been doing at Framingham and BUADRC, and then we're trying to deploy this globally. Where are we trying to get to? We're trying to get to this concept of digital biomarkers. Lots of people use those words now. In fact, when I first started using them, I was told I wasn't allowed to use them, but now they become the heyday, except for I think we're using it incorrectly. Right now, we're using digital biomarkers. It's just a better version of what we're already doing, and that's not good enough. What we have to realize is we're going to have to take these multi-sensor digital streams. We're going to have to work with our data science AI colleagues in terms of how to analyze them. And we're going to have to come up with very different kinds of patterns. They're going to be very dynamic. They're not going to be single threshold, single measure, right? It's going to be this constant flowing dynamic flow of information. The combinations are continuously going to change, and nonetheless, this is how we're going to get to accurate assessment. And that's going to move us then into the realm of doing virtual clinical trials and into clinical care. So I'm going to stop here by just telling you to do all this work, you actually have to bring lots and lots of people together. And what we have to be very careful in research is that we're not prescriptive as to who we think those could be that we can work with. So thank you very much.
Hi, thank you. Thanks for the invitation. I'm a vascular neurologist over on the other campus. It's always great to come visit on this side near the river. So I'm going to be talking about blood biomarkers for Alzheimer's disease and related dementias. As recently as 10 years ago, Alzheimer's was categorized or binned mainly into Alzheimer's disease, vascular dementia, and then everything else. That's problematic because, as we know now, there are so many underlying risk factors, social determinants, and genetics that underlie the different types of pathology that you can get. And then only at the very end, you have these clinical presentations with different types of dementia. And so the big problem is that all of this different pathology is happening over years and decades. And it's been problematic for our clinical trials because by the time we are treating patients, when we recognize they have a syndrome, it's often too late. So towards this, NIH and NIA have been trying to put together better definitions for different types of dementia, focusing on Alzheimer's first and trying to get a biomarker definition towards a biological definition of Alzheimer's disease. And they put together this that would incorporate amyloid disease protein deposition, tau, and neurodegeneration. And so this came out in 2019 and was very controversial, but from the get-go, it was noticed that part of the problem is that to make these diagnoses, you have to have cerebrospinal fluid, you have to have PET neuroimaging, CSF being collected by lumbar puncture, which is invasive and potentially can have complications, and then PET being kind of expensive. None of this can really be done at scale and requires heavy resources, as Rhoda was speaking about before. So the thought is that in the future, we're going to be able to use blood-based biomarkers and neuroimaging, less expensive neuroimaging, to be able to start catching dementias that occur preclinical, so before they turn into a syndrome. So part of the work that we've been doing over the last few years in the Framingham Heart Study, where it's part of a research group that I'm with, looked at the tau protein, a protein that forms part of the structure of neurons. And it's been found that you can measure that tau protein in the blood. And what Dr. Passe was able to show was that this persons in the Framingham Heart Study who at baseline had higher than the median level of t-tau in their blood compared to those with lower than the median level of t-tau were more likely to progress to all dementia and also to Alzheimer's dementia. And another biomarker, and this time being researched in the Alzheimer's Disease Research Center, is neurofilament light chain. It forms the neuron cytoskeleton and is found in the plasma of the blood. And here they were able to show that baseline NFL predicted declines in the mini-mental status exam, declines in verbal fluency, and declines in logical memory. And that was led by Dr. Alasko. And then Dr. Stein and our group we were all able to put together a study using pathology from the brain bank from autopsy cases, where they stained for neuropathology. And what you can do is you can phosphorylate tau, a subspecies of tau that's more specific for Alzheimer's disease, and actually predict during life what pathology will show on the brain, even in people without mild, moderate, or severe cognitive impairment. And it also was able to predict progression to higher Brock stage, which is to show the spread of that abnormal protein in the brain. And another class of protein you might measure might be inflammation, markers of inflammation. And so in a consortium group that we're working with CHARGE, which are population-based studies like the Framingham Heart Study, and then Mark VSID, which is funded through the NIH as well, we were able to study glial fibrillary acidic protein, which showed that with measuring that at baseline, it predicted both all dementia and Alzheimer's dementia over a period of many years, up to 5 years. Similarly, using kind of the same cohort, looking at YKL40, another inflammatory protein from astrocytes, Dr. Passe is leading a study showing that it can, in cross-sectional analyses, can predict total brain volume and also worsened global cognition. We're also looking at dementia in the framing of our study using that YKL40 biomarker. Just to see how widespread this can be used, I'm not part of this research group, but Dr. Stern has this study on chronic traumatic encephalopathy or repetitive head injuries, and they're also looking at these inflammatory biomarkers to help predict cognition in people with head injury. So, in summary, we are now using these studies to be able to not only just study them in retrospective studies, but look at real-world performance and implement them at the bedside. And this is really important because now we know so much more about the kind of pathologies that occur in the older brain. And so Mark Visid is helping to look at reliability and instrumental validation of these tests, both blood biomarkers and imaging biomarkers. And then finally, really at the bedside, we're recruiting patients with stroke at BMC and 30 centers across the US to then measure these things like the blood biomarkers to see if they go on to develop dementia after stroke. Thank you.
Thank you to the organizers for strategically placing my talk right after the neurologist spoke. I'm going to talk about how AI tools can be built to actually meet the standards of the neurologist. So I'm nervous. So our lab is located in the Met campus, and we mainly have two kinds of students. One of them is getting a PhD in computer science, and the other person is actually getting an MD degree in the School of Medicine. And we sort of put them together in a room and then ask them to work on something interesting, and they come up with some magic. And basically, the reason we want to work on this is because of a problem that I think some of you probably know. I think maybe Hugo, you would agree with it. There is actually a shortage of experts. There is a shortage of experts in terms of actually diagnosing the disease. Dr. Rodo mentioned about this forward-thinking initiative about thinking about precision brain health, which I think is amazing. This is what we need to do. Statistically speaking, if a patient actually if a patient is sick, they spend maybe an hour or two in the hospital. For the remaining thousands of hours, they're actually staying at home, right? Which means we need to think about those forward-thinking initiatives to monitor brain health over time. But in reality, when the patient is seeing the doctor is very overwhelmed because there are so many people, the shortage of experts, and I think the statistic here says that the US could use about 11% more neurologists today, and then the expectation is that it's going to go to about 19%. So which means we need experts. But if I talk to an MD student who wants to research, they don't want to necessarily become neurologists. I think there's a very small percentage of MD students who join a class of 150 actually want to be neurologists. We talk about financial benefits, but I think there are many other reasons in terms of burnout, etc. So how can we help? How can a computer scientist, working in School of Medicine help? I think what we are trying to do is we're trying to build these AI-based approaches that can hopefully one day serve as screening tools to assess dementia. And our goal is to not just take data, our goal is to take data in its native format without any processing by the physicians, which means we want to basically grab the data from the electronic health records, whether it's data coming from imaging, clinical demographic data, patient history, cognitive testing, emerging digital data such as voice recordings. We want to process all of these things, and then we want to build AI approaches that can hopefully assess dementia that one day could be as good as what the neurologist can do today. We focus heavily on comprehensive validation. So we not only just build methods, we want to make sure that these methods are validated because that's, I think, a very important thing to do. This is just an overview of the recent work we did. We start off with data. And I think when we say data, we're not just collecting data from one center, we're collecting data from multiple different centers. So which means right off the bat, we're talking about data in different formats. Some of the data is missing, some of the data is coming from a different language. So we want to figure out a way to sort of harmonize all that information. So we start off with basically building a set of computation routines that can automatically get all this data, then make this data available for machine learning. And once we build those kinds of pipelines, we then start building these multimodal deep learning approaches to sort of fuse information coming from all kinds of features. You see that demographics, functional assessments, neuropsychological tests. These are all the things that are actually done at the same grade at DLC. So we want to grab all that information, we want to integrate all that information, and we want to sort of ask a simple question, can you assess this person's cognitive status, whether it's normal cognition, whether they have mild cognitive impairment or dementia. And once you have dementia, you want to also understand what kind of dementia. Is it because of Alzheimer's or is it because of something else? And then once we build the models, we want to make sure these models are validated computationally. So we train it on one single cohort and we sort of test it on all these other cohorts. And once we do the computation validation, we sort of seek experts like you to sort of validate these models. So we gave about 70 different neurologists about 100 cases, and then we asked them what they think about those patients. And then they compared our model performance on those same 100 cases and sort of validated how our model was at least as good as the average neurologist. And once we did that, we also talked with a few radiologists who basically also looked at these cases and looked at imaging data. And then they identified regions in the brain that are indicative of Alzheimer's and those with other etiologies. And we again compared our model with the expertise. And finally, on a few set of cases on people who actually died, their brains were donated. And we sort of looked at how model predictive regions identify those disease risk of pathologies in those specific regions. So I think there are 3 layers of validation we did, and that is, I think, sort of really helping us sort of build, hopefully one day, tools that can be essential for dementia. Thank you.
Okay, hi everyone. Thanks for having me today. My name is Claire and I'm a postdoc in Dr. Kieran's Aphasia Research Laboratory. And what I'm going to talk about today is a recently initiated F32 project that I'm doing under the mentorship of Dr. Kieran and Dr. Kolachalama. So it fits in nicely with what people have been presenting. So I'm specifically going to zoom in thinking about speech as a biomarker. And I think, as some of the speakers have alluded to, connected speech is a really cool opportunity because it's a window into many different cognitive and linguistic facets. And so it gives us the opportunity to look at quantifying different measures of connected speech and using that to see what sort of deficits a patient might have. So the question becomes then, what kind of measures are we interested in looking at? And that really depends both on the population and the suspected deficit in an individual. And so today I'm sort of going to narrow in the focus for my project, looking at speech fluency in post-stroke aphasia specifically. So fluency in aphasia, and it's an important concept, first and foremost, because it's salient to patients. How fluent your verbal output is mediates how you interact with people in your everyday life. And this is, of course, important to people. It's also important from a diagnostic perspective, because there's a tendency in speech pathology to bin individuals at the broadest level into whether you have fluent or non-fluent aphasia. And this has downstream implications in terms of devising rehabilitative targets as well as prognostication in some cases. But of course, because as we saw on the previous slide and what other speakers have been saying, connected speech is complicated. So it involves, for instance, finding words, stringing them together into sentences, and also articulating them motorically. So as you can imagine, because it is a complex construct, there's not really a consensus on what the best assessment approach is. And so typically the current approaches tend to fall into one of 2 categories. Either it's a Gestalt clinician judgment, oh, this person sounds fluent or sounds non-fluent. And as you can imagine, that tends to be unreliable from person to person, even among experts. Or there are these more detailed quantitative approaches, but they often rely on this very time-consuming manual transcription that requires either an expert, either a clinician or a linguist or such. And so where our project sort of fits in is thinking about, can we import some of the automated speech analysis tools that have been used successfully in other patient populations and test those in post-stroke aphasia? And specifically, is there a role for potentially machine learning approaches to help us identify which features to really hone in on that carry the most predictive value? So just before the next slide, we'll get into more details about the project, but just big picture overview. We know from prior work that there's promise for what I'm going to call quantitative temporal acoustic features. So these are features where we're not concerned so much with what the person is saying, but how they're saying it. So zooming in on like the rate and rhythm of speech. How much total speech is there? Where are people pausing relative to different syntactic boundaries and things like that? So all things that you can get from a continuous speech stream with minimal processing. So we know from work that we've done and that others have done, we have validated approaches for this kind of automation. And we know at the group level that they can differentiate fluent and non-fluent aphasia. But where my F32 project is trying to go is taking that a step further to the level of individual prediction. And so I just want to highlight, of course, my mentoring team, Dr. Kiran and Dr. Kolichlama here, who you'll hear speak, and then Dr. McWhinney at Carnegie Mellon as well. So this is a two-part project. The first is asking the question, can we use these features to meaningfully distinguish between fluent and non-fluent aphasia? And more than that, which features are actually going to be the best performing ones? Because that will help us narrow in and eliminate redundant features, for instance. And then the second aim is really coming from my clinical background, which is, okay, we know which features may be most important in terms of the performance of the algorithm, but we also want to understand what these features mean and what are the underlying speech and language and cognitive constructs. So for instance, someone has slowed speech. Is this a problem of motor speech? Is it a problem with the retrieval of words? Is it a problem with syntax and so on? So it's actually trying to understand the underlying reasons behind these surface speech characteristics. And that's ultimately the knowledge that we want to translate back to practicing speech pathologists. All right, thank you.
Okay, I am the last speaker, and I'll also try to close out today's talks. I want to talk about a computational model that we've been building for the last few years. It's called Bilex, and it is meant to design and simulate a person who's bilingual and how they process words and speak words in two languages. This is what it looks like. It basically has vector representations of words like dog and what they sound like and what the features of a dog are. And what it can do is simulate a person who's bilingual. And on the left-hand corner, you can see a person who's extremely bilingual, and the map looks very rich. And on the right-hand corner, you can see a person who's bilingual, but only exposed to one language much later. So they're not very proficient in that language, and the map looks quite not as rich. It looks a little sparse. So what you're going to see on that top panel there is how the model tries to learn this language over time, and just pay attention to the semantic map and the other two maps. And one of the languages is being learned later and later in life. So what we have done is we've developed this model, and we call it a pre-stroke model because it does a pretty good job of pretending to be a bilingual person. And then we took this model and introduced noise into it, and we tried out different types of noise models. And we either put it in where the meaning of the words are or where the words are produced, and we call this the post-stroke model. But then what we did with that model is we took it and tried to rehabilitate it. So we tried to see whether it actually can recover the language it had lost. And the key thing about this is that the model can actually can simulate its own recovery in two languages, in English and in Spanish. So that's what those lines show: improvement in English and improvement in Spanish. And this paper that we published showed that the model actually does a pretty good job of simulating what human patients actually do when they receive speech therapy. So this is sort of what the prescription is now. The model can tell you beforehand which language is going to improve more based on the amount of damage you give it. And it can actually tell you, based on that improvement, you should probably treat in one language versus the other, because it knows what the outcome is going to be beforehand. So this is all very exciting, because now we're almost done enrolling all our patients into this double-blind randomized controlled trial with 48 patients who are only receiving therapy in one language. It's either English or Spanish, but the language that they're receiving therapy in is being prescribed by this computer model and telling us which language is more improving. So this is where we're almost done. We're going to analyze the data very soon, but in the meantime, we're now extending this to dementia. And the goal here is to see whether we can actually predict decline in patients in this naming process over time. So assuming that you start with the diagnosis in year 1, can you see where the patient is going to end in year 2, 3, and 4? And this is not a simple problem to solve, because in stroke, we're actually trying to simulate recovery. So there's a damage, but we want to show improvements over time. But in dementia, we actually want to show the slow decline over time. So we've been trying out different types of noise models, but the noise has to be degenerative. So in this particular noise model, we're trying to introduce, just take out parts of the semantic map progressively over time. In this sort of, lesion model, stroke dementia model, we're trying to damage the input that the brain receives. So it sort of feels fuzzy and it doesn't get all the information that it's supposed to process. And in this particular damage model, we're taking out the connections. It's a lot like what we would expect to do in stroke, but now the connections between your image of the word, like what a dog is and how to say it, are being progressively lost over time. So these are all different options for us to test out, but what's been really promising so far, at least, is that the model does a pretty good job of simulating patients with primary progressive aphasia over time. And so what you see in this last figure here is actually data in the dots are actual human patients who've been tracked over time, and you can sort of document the decline in their naming abilities, and the lines are all the model simulations of that. And at least in these two patients as samples, the model does a pretty good job of predicting where the patients are going to end up over time. And this is really potentially important because even if we come up with new treatments for people with primary progressive aphasia and dementia, we don't know what the rate of decline is going to be, but the model allows us to make precision-based predictions of where the patient will end up depending on where they started off. So that's what we're trying to do right now. So I'll stop. I think I'm done early, actually. I timed my talk for 4 minutes, so I think we're good. I'd like to close out having Gloria come back up and just do a quick round of thanks. As you can see, the topics are fairly broad, but there's something that connects all of these together, and I hope you could see the connection. And I think the way we sort of planned this was at the end, you can see the value it has for patients. And I will not reduce the emphasis of what we're trying to do here. I recognize at least one stroke survivor in the audience here today. Thank you for coming. And they're here, they're watching us, they're waiting for us to come up with new discoveries. And I think it's really important that we take this, like what we're doing seriously and make a difference. So thank you for coming.